right, this is the last section in chapter 3. So this one is on nucleic acids. So these are essential macromolecules that allow the cells to survive. It carries the cell's genetic blueprints. So this is the instructions that allows the cell to function properly. This is the instructions that tells the cell what proteins to make in that primary sequence. It does other things besides that, but that's like most of what it does. There are two main types of nucleic acids. There is DNA and RNA. DNA is the genetic material found in all living organisms. RNA is more of a transitional molecule that we find in protein synthesis and regulation. So we use that to make proteins. And we'll talk about all the different types of RNAs when we get to the DNA RNA chapter. So in this chapter, we're just kind of introducing those molecules. So it is a monomer. That monomer is a nucleic acid. DNA and RNA have 4 different monomers called nucleotides. That nucleotide includes a sugar, a nitrogenous base, and a phosphate. It's this nitrogenous base that's different. We have 4 different nitrogenous bases in DNA and 4 in RNA. So, and again, it has, you see all the nitrogen, which is why we call it nitrogenous base. The sugar is different in DNA and RNA. One is, they're both ribose sugars, just one does not have a hydroxyl group on it. And so we call it deoxyribose. So they both have a 5-carbon sugar. We usually name those by that being carbon 1, 2, 3, 4, and that's our 5th carbon. So here are the different nitrogen bases that we have. We have what are called pyrimidines and purines. The pyrimidines are single rings. The purines are double rings. I always remember that the longer word is a smaller molecule, and then the smaller word purines is a bigger molecule. It's 2 rings. That's how I always remember which is which. So we have our cytosine, thymine, and uracil versus our adenine and guanine. And we'll talk about base pairing and all that good stuff when we get into our RNA and DNA. But here's our different sugars. You can see right here in the ribose. I have to be careful because it sends me to the link. That has that OH group. It is missing in our deoxyribose. So there's our difference in our nucleic acids. Our nucleotides. Nucleic acid would be RNA versus DNA. It's a chain. They're chains of nucleotides. In DNA forms a double helix. RNA is generally single-stranded. We will base pair between our pyrimidines and our purines with hydrogen, and we can see those base pairs here and here. Again, we'll talk much more about this later. But DNA is, like I said, a double helix. And we can see that we have our deoxyribose down here. The differences between DNA and RNA, again, the function of DNA is it carries it's the master copy of information, where RNA is involved in protein synthesis. The location is the DNA stays in the nucleus for eukaryotic cells. Of course, we don't have a nucleus in our prokaryotes. In RNA, we will make the RNA in the nucleus, but it will leave out the nuclear pores and go into the cytoplasm. The structure is a double helix for DNA. Like I said, RNA is usually single-stranded. The sugars, deoxyribose versus ribose. And then our pyrimidines, we use C and T. And in RNA, we do not use thymine, we use uracil instead. So RNA and DNA both use cytosine, adenine, and guanine. The biggest difference is right here with our thymine versus our uracil. And then we talk about our central dogma. It's called the central dogma of molecular biology, and that is that we go from DNA to RNA, from RNA to protein. We talk about that process going from DNA to RNA. We're talking about transcription, like to write a copy. We're transcribing a copy of DNA in the form of RNA. And then we're going to take that RNA and translate it into a protein. And so we will specifically be learning how to do this again later, all in chapter 10, which it has to do with transcription translation. We'll get into DNA replication as well. And so now we just have our conclusion. We talked about all of our living things are carbon-based. We are carbon-based organisms on this planet. We talked about functional groups. We spent some time on carbs. We talked about lipids, proteins, nucleic acids being the four major classes of biological macromolecules. We did those labs, or will be doing those labs in our lab. They are very important. Most of these biological macromolecules are made up of monomers, which are single repeating units. Not all. Remember, lipids don't really follow that pattern. And then we talked about our dehydration and hydrolysis reactions in order for either a catabolism or an anabolism. And then, of course, we also talked about our carbs. Just a little more detail, our monosaccharides, our disaccharides, our polys. We talked about what their functions were. We talked about the different classes of lipids, whether they're fats, waxes, phospholipids, which are important in the cell membrane, and steroids, which are used for cell signaling. And then we also talked about, we spent a lot of time on proteins, their different sequences, primary, secondary, tertiary, quaternary, how that relates to their function, and denaturization, things that can cause denaturization. And then lastly, we talked about nucleic acids. So this chapter I know has a lot of information, and it's usually one of the harder ones to grasp. So please spend plenty of time going through this. I would also be going through it with your lab. The lab will help too, as the labs are meant to help enforce the learning in the chapters. So hopefully gives you more of some hands-on, since you were actually handling these different biological macromolecules in the lab and testing them. All right, hopefully that was helpful, and I will see you in the next video.